Hey there, everyone. So let's go ahead and pick up where we left off in Chapter 10 with muscle contraction. This is where we left off in Part 1. So basically, we were just kind of covering all of the necessary steps that occur in the excitation-contraction coupling of skeletal muscle. And what we're eventually going to see, these steps are going to really mostly be the same for cardiac and smooth muscle too. So just a quick little recap here. In order for a skeletal muscle to contract, we need stimulation from a somatic motor neuron. So it releases its neurotransmitter of choice, which is acetylcholine. Acetylcholine binds to nicotinic receptors here in the neuromuscular junction. Those nicotinic receptors, as we've discussed many times before, are always excitatory, so that will depolarize the muscle fiber. The action potential will be conducted down the length of the transverse tubule. That will lead to the opening of voltage-gated calcium channels in the sarcoplasmic reticulum. Calcium will start flooding out into the sarcoplasm, where it will bind to troponin. Troponin will move that tropomyosin out of the way, uncovering the myosin binding sites on the actin, and that allows the myosin, which is cocked back and ready to go, to grab onto the actin and move it along to give us our power stroke, which we need in order to shorten the muscle, shorten the sarcomeres, and get our muscle contraction that we so desire, right? So now that we've talked about all the requisite steps that are involved in muscle contraction itself, let's talk about getting things back to a relaxed state. Once you have your contracted muscle, let's get things back to where it was initially. So the first thing we want to tackle here is we want to make sure that we understand, in order to get a muscle contracted, what did we need to do to get there, right? So, number one, we needed acetylcholine in the neuromuscular junction acting on those nicotinic receptors. Number two, we had to get calcium out of the sarcoplasmic reticulum into the sarcoplasm where it could bind to troponin. And then finally, we needed an energy source, right? So in order to get those myosin heads cocked back and ready to go, we needed ATP molecules, because that cocking back into the high potential energy state cannot happen if there is no energy there. So really, these are the three major things that we need in order to go from relaxed muscle to contracted muscle. So now if we look at this through the lens of going back to a relaxed muscle, we're basically going to push the rewind button here on these different things. So for acetylcholine, once we decide to relax a muscle, there are no longer going to be any action potentials traveling through that somatic motor neuron. So no more acetylcholine release, but that doesn't really do anything for the acetylcholine that has already been released into the neuromuscular junction, right? So we need to get rid of that. So we've covered this before, but for any neurotransmitter, there's going to be an enzyme in the synapse that is going to break down, degrade, or otherwise recycle that neurotransmitter to get it out of the way. And in this case, for acetylcholine, that enzyme is called acetylcholinesterase. So the muscle can only truly relax once that enzyme has cleared out all of the acetylcholine and the muscle fiber can go back to its resting membrane potential. Okay, so at that point, no more nicotinic receptors activated, no more acetylcholine. The muscle fiber goes back to its resting state. So with no more action potentials traveling through the T-tubules, those voltage-gated calcium channels in the SR membrane are going to close. So no more calcium leaking out of the SR. But we need to do something about all of that calcium that has already escaped, because we already have quite a lot of the calcium in the sarcoplasm binding to troponin, keeping the muscle contracted because tropomyosin's been shifted out of the way. So if we want to go back to a relaxed state, we need to clear out that calcium so that tropomyosin can reconfigure itself to block those myosin binding sites. So the way we're going to get calcium back out of the way is we want to use active transport to pump any of that calcium in the sarcoplasm back to where it came from, back inside the sarcoplasmic reticulum. So essentially what we're doing here is we're just resetting things back to the way they were before we actually got the action potential. And then number three here is kind of interesting because We're not necessarily going to do anything specifically in terms of reversing course here, but it is worth mentioning that for now, and this will be important in a little while, if we did get to a point where there was no ATP, the myosin heads could not be able to cock into place to generate power strokes. Specifically, as we're going to see, if you don't have any ATP, you're actually not going to be dealing with a totally relaxed muscle. It's actually going to be quite the opposite. So this is the one thing, number 3 here, that doesn't quite jive with the other two, but we will make some sense of that here in just a little bit. So ultimately, whether you're talking about muscle contraction or muscle relaxation, the whole point of it all is to increase the concentration of calcium in the sarcoplasm. Basically, the way to think about this, if you want to think about it in a very simple way, if there is calcium in the sarcoplasm, the muscle is going to be contracted. If there is no calcium, the muscle will not be contracted. As long as there is enough energy available in order to get the myosin heads cocked back, it really comes down to just that. So muscle contraction is about making calcium available. Muscle relaxation is about putting the calcium back in the sarcoplasmic reticulum. So here's that interesting thing about ATP that I was talking about before. So what we're going to talk about here is an interesting case study on rigor mortis. I'm sure you probably have a pretty good idea of the type of people that experience rigor mortis, and unfortunately for them, it's dead people, right? So before we actually talk about the rigor mortis, we are actually going to use this as an opportunity to make sure that we understand what we're going to call here the cross-bridge cycle. So this is something that we've already kind of explored already, but let's take a look at it in a little bit more detail on a step-by-step basis. So if you look at this diagram right here, it has this particular slide right here as kind of our starting point. We're not actually going to start here. We're actually going to start back here. This will kind of be where we assume our starting point is here with number 6. So what we're looking at here is a myosin head that is not quite cocked back into its high potential energy configuration. And you can also notice that it is not bound to the actin thin filament. So definitely we're dealing with relaxed muscle at this point. So you'll notice here that this myosin head is actually bound to a molecule of ATP, a high energy ATP molecule. So what this myosin head is going to do is hydrolyze that ATP, harness the high amount of energy in there to cock itself back into that high potential energy configuration that we've talked about. So going from 6 to 1 right here, you can see that that is the case. We change the shape and conformation of that myosin head, and you can see it's all rearing and ready to go here. And by now, we no longer have ATP. We have the much lower energy configuration, ADP. So you'll notice at this point, even though we have already spent that ATP molecule and cocked the myosin head back into its high potential energy configuration, you'll notice that it is still not bound to actin. Those the myosin heads and the actin thin filaments are still separate from each other. So we have not yet formed a cross bridge. And even though it is not illustrated in this diagram, we understand now that the reason that that is the case is because currently in this picture at number 1, tropomyosin is blocking those myosin binding sites, and we're not going to get a cross bridge until calcium gets released from the sarcoplasmic reticulum. So as long as you understand that, you're in great shape so far. So we have not yet formed a cross bridge, but moving from 1 to 2 here, we'll just go ahead and assume that we've gotten our calcium and troponin has moved tropomyosin out of the way. So here you can see that myosin is now able to form a cross bridge with the actin thin filaments. As soon as that binding occurs, we get our power stroke down here at number 4. So this black arrow you see here is meant to indicate that the myosin head shifts itself forward and moves that actin filament along. We shorten the sarcomere and get our muscle to contract. So that covers everything from going from relaxed muscle to contracted muscle. So now from the perspective of the cross bridge, meaning the myosin and the actin, the last thing we need to cover here is how do we go back to relaxed muscle. So for now, go ahead and pretend like this red X right here is not there. So just pretend that's not there for right now. So currently, we have contracted muscle because myosin is tightly bound to the actin. If we are going to go back to relaxed muscle, we need that myosin to let go. And the way that it's going to let go is it is going to exchange that low energy ADP molecule for a new high energy ATP molecule. This exchange allows myosin to release itself, to dissociate itself from the actin, and we go back to our starting point here at number 5. So now that we have established a new affinity with a new ATP molecule, we go back to where we started with myosin in a low energy configuration, not forming a cross bridge. And that's the exact way it's supposed to go. Muscle contraction is all about this cross bridge cycle going again and again and again and again. Every time you go through the cycle, you require a new ATP molecule. And since there are millions, if not billions, if not trillions of myosin heads in every skeletal muscle fiber, you can appreciate how keeping a muscle contraction going is going to eat up a lot of ATP energy. It's a very energy consuming process. So now let's look at this through the lens of rigor mortis. You're probably aware that rigor mortis is basically what dead people go through not long, maybe an hour or two after they die, in which their muscles stiffen up and basically they become rigid as a board. So the idea behind this we can actually explain using the cross-bridge cycle here. When a person dies, their metabolism stops altogether. So their cells, their mitochondria, are no longer synthesizing ATP. ATP is going to be used up and depleted very rapidly. So we get to the point as we go through these cross-bridge cycles in which we run out of ATP, meaning we get to this point here where we cannot progress past that red X right there. So when those cross bridges get to that point, we basically lock the muscles in a configuration in which they are contracted. And this helps us to explain why a dead person who is in the stages of rigor mortis feels stiff as a board. It's because their muscles are locked in a contracted state. Now, certainly this is not a permanent process for several reasons, including bacterial putrefaction, breaking proteins down, and other reasons. This will kind of abate after several hours. But if we, again, use this as an opportunity to instruct ourselves on ATP and the cross-bridge cycle itself, we can kind of look at this in more detail than what the previous slide was showing us. Now we can actually see tropomyosin and troponin here. I had mentioned this in part 1, but make sure you understand that myosin head will use ATP to cock itself back into place even before calcium becomes available. I said it last time. With all of your relaxed muscles right now, so if you just relax your muscles right now, all of those myosin heads are cocked back and ready to go. That just because a myosin head is cocked back and rearing to go does not mean the muscle is contracted. What has to happen is that cocked and ready myosin head has to bind to actin, and we need calcium and troponin and tropomyosin in order to do that. So the head will stay cocked until those actin binding sites are uncovered by troponin and tropomyosin. And then once the filament sliding occurs, once we get our power stroke, the myosin head will have to release its ADP and find a new ATP molecule in order to get that relaxation to occur. So moving ahead, one of the most important discussions that we're going to have in this second half of Chapter 10 is on muscle fatigue. You know what muscle fatigue is all about. Everyone, everywhere has experienced muscle fatigue before. But we still have a little ways to go before we can really have a good, clear discussion on what muscle fatigue is, and we want to be able to understand that. So we are going to cover a couple of topics before we get there. Number 1, we want to look at muscle contraction, as we just did, through the lens of the energy requirements. We understand now that muscle contraction and muscle relaxation require this constant influx of new ATP molecules. So you may have heard me before mention that ATP is kind of like currency. It's kind of like the cell's money that it deals with. In the real world, if you want to get something done, you've got to have money to spend to get it done, right? But money is something that can be accumulated and stockpiled. You can measure your personal financial wealth based on how much money you have in the bank, how much you have invested, and so on. ATP does not really work that way. It's not something that you can store up and stockpile. Rather, when your mitochondria generate a whole bunch of ATP, that ATP is all going to get used up pretty much right away. So it's not really something that we can kind of set aside and use later. So your muscle fibers actually have a system for storing up and stockpiling energy independent of ATP itself, and this is called creatine phosphate. So creatine phosphate is kind of like ATP. It's a very high energy molecule. If you look here, we actually use ATP in order to create creatine phosphate. We use an enzyme called creatine kinase to attach one of those high energy phosphates directly onto creatine, and we generate a high energy creatine phosphate, which, like I said, can be stockpiled and can be accumulated. So the way that this works, and you might be thinking this is kind of weird, we use ATP to make this anyway, so it's almost like, what's the point? Well, let me explain this, and let's see if maybe it makes a little bit more sense afterwards. So in resting muscle, meaning you're not really doing anything overly strenuous, your ATP consumption, oxygen consumption, and so on in your skeletal muscle is not very high, right? So energy demand is relatively low. So even so, your mitochondria are going to be making a lot of ATP. So if we're making more ATP than we need currently, we can take some of that excess ATP and convert it into creatine phosphate, which we've already established can be saved for later. So if we build a nice stockpile, a nice cache of creatine phosphate, later, once our muscles do become active, whether we're working out or doing some other type of strenuous activity, we're going to run out of naturally produced ATP very, very, very quickly. So it's nice to have this creatine phosphate that we can tap into to give us a nice burst of ATP energy when we really need it. But regardless, whether you're talking about ATP or creatine phosphate, your muscle fibers will still need to make ATP by utilizing the molecules that we get in the food that we eat, right? So we're talking carbohydrates, proteins, fatty acids. So basically what you're looking at here is we're basically assuming that our muscle fibers energy is coming from the glucose that we get from carbohydrates, although you can do fatty acids and proteins too. But we'll just go ahead and assume we're talking about glucose here. So glucose from the blood, we've talked about all that with glucagon and insulin and all that good stuff. Glucose from the blood will be delivered to skeletal muscle fibers at a pretty steady rate. But there will be cases when you're really working out very strenuously when the energy demand in a muscle fiber actually outstrips the ability of the blood to deliver those food molecules to the muscle fiber itself. So basically, our ATP demand is going to exceed our ability to make ATP. One is faster than the other. So our need is faster than our ability to make good. So in these cases, if we're not getting a fast enough delivery of glucose, we actually have a storage unit for glucose already contained within the skeletal muscle. And this is something that we've talked about before. This is called glycogen. It's also present in liver tissue. And you will recall the hormone glucagon actually can stimulate what is called glycogenolysis, the breakdown of glycogen. As a reminder, that is a long polysaccharide, a long polymer of glucose molecules. If we start hydrolyzing glycogen to free up individual glucose molecules, now suddenly our skeletal muscle has something to feed on, something to make ATP out of. And you'll recall way back in chapter 17 when we talked about hormones, you'll recall the hormones epinephrine from the adrenal medulla, glucagon from the pancreas, and cortisol from the adrenal cortex all can work together in synergistic fashion to stimulate glycogen breakdown in skeletal muscle. That way we have a quick burst of energy to get through our workouts or whatever else. But, as we're about to see, all of this is going to kind of be for naught if we cannot deliver a steady stream of oxygen to that skeletal muscle as well. As we are going to see, our ability in the mitochondria to make a lot of ATP for the muscle fiber to use is going to greatly depend on whether oxygen is available or not. So, if oxygen is readily available, our mitochondria will utilize cellular respiration. And as we've discussed way back when, for every 1 glucose equivalent, about 36 molecules of ATP are going to be made. So that's quite a lot of ATP. But if oxygen is not readily available, meaning if we are using a lot more oxygen than we are delivering, then we cannot go through cellular respiration. And instead of using the mitochondria, we are instead going to use lactic acid fermentation, which takes place in the cytoplasm. So lactic acid fermentation is kind of a problem. It is basically just glycolysis and the 2 ATP we get out of that for every glucose equivalent. And then lactic acid fermentation doesn't really make any more ATP. It basically just resets things with NAD so that we can go back and do another round of glycolysis. So the problem there is twofold. Number one, you are not making enough ATP to really support the energy demand of the muscle fiber. And as we are going to see in a little while, that is going to play an important factor in muscles getting tired, muscles fatiguing. And number two, all that lactic acid that builds up kind of plays a role in getting our muscles cramping, basically giving us that sore muscle feeling that kind of makes us not feel very good. So, if we do work out very strenuously such that our muscle fibers simultaneously run out of ATP, run out of glycogen stores, run out of creatine phosphate stores, if any of those things occur, we say that the muscle fiber is in a state of oxygen debt, the same way it works with financial debt. If you're in debt, you have to pay it off in order to get yourself out of debt. So the idea here, the way your muscle fibers get out of oxygen debt is, number one, you need to stop using them so you can kind of get yourself stabilized. If you're in financial debt, you want to stop spending money, right? So you got to stop spending before you can really start thinking about paying back. So once your muscle fibers are no longer being overutilized and no longer blowing through your stores of ATP and oxygen, you are going to be breathing very, very, very heavily, something that we will talk about in Unit 5 here in about a month. So you'll be breathing very heavily, getting lots of oxygen into your lungs, lots of oxygen into your bloodstream. So you will be basically playing a waiting game in which your muscle fiber is no longer using a lot of energy, so it can use that time to catch up, to resynthesize glycogen, resynthesize creatine phosphate. Once we get those levels back to where they were before we got into oxygen debt, we can declare ourselves basically out of the oxygen debt at that point. So another set of things that we need to really go over before we start talking about muscle fatigue is we want to say a few things about the level of control the central nervous system has over muscle contraction. And you're probably thinking that, okay, I have a pretty good understanding of this. Somatic motor neurons, action potential, we release acetylcholine into the neuromuscular junction, we activate nicotinic receptors, and boom, everything happens from there. And you're not wrong. But that was under the assumption that one somatic motor neuron has control over one muscle fiber at a time. And as we are going to see here, that is not the case. So let's dive right in here. So take a look at this picture right here on top. So you're seeing a section of the spinal cord with your pair of spinal nerves protruding out on either side. Look down here on the right side of that section of the spinal cord in the gray matter in the ventral horn. What you're seeing there are two cell bodies of two somatic motor neurons. So very quickly, let's follow the axon of this somatic motor neuron right here, the one that's kind of on the bottom there. So if we follow that axon, everything looks normal so far. Once we get down past the myelin sheath, look at what happens to the axon there. It is diverging and it's forming kind of all these crazy little branches here. Specifically, look at the axon terminals. Let's count up those axon terminals right there. So we've got 1, 2, 3, 4, 5, 6, 7, looks like 8. There might be a ninth one there, I can't really tell. So let's just say 8. So this one somatic motor neuron has 8 axon terminals. And what we're going to see here is that every axon terminal synapses with one singular muscle fiber. So this somatic motor neuron, which has 8 axon terminals, that somatic motor neuron controls simultaneously 8 different skeletal muscle fibers. And then if you look at the other somatic motor neuron, you can see the same is true. If you count all these up, I believe it's 10 axon terminals. So that one, that somatic motor neuron has 10 muscle fibers under its jurisdiction. So this is kind of crazy, right? So we actually have terminology that we use for this. Each one of those somatic motor neurons, along with all of the muscle fibers it has control over, each one of those is called a motor unit. This is basically the way it works. So if in this picture that we have right here, we have 2 different motor units, one under the control of one somatic motor neuron, the other under the control of the other. So if one of these somatic motor neurons here, let's say the one on the bottom here, if that one fires an action potential, that action potential will spread to all of the axon terminals, and we will get acetylcholine release from all 8 or all 10, depending on which neuron that you're talking about. So if it's just one of these somatic motor neurons firing an action potential, we will get these on bottom here, these muscle fibers contracting, but not the muscle fibers in the other motor unit. If we want all of those muscle fibers to contract, we would need both somatic motor neurons to fire action potentials at the same time. So this leads into a discussion called recruitment. Recruitment refers to your ability to use the central nervous system to activate progressively more and more and more somatic motor neurons in order to get recruited more and more motor units and more muscle fibers. So if you, say as an example, lift something that is very light, you don't need very many muscle fibers to do that. But if you switch over to something that is much heavier, like for me, I'm using my microphone here, this isn't exactly a light object, so I needed to recruit additional motor units in order to give me the strength and contractile force I needed to pick it up. So we are just about ready to have our discussion on muscle fatigue. Just a couple more things that we need to say about muscle contraction and the nervous system here. So when a somatic motor neuron fires one single action potential, so just one action potential, no others after that, the muscle fibers in the motor unit will receive the input and they will contract once. And that one contraction is what's called a muscle twitch. So a muscle twitch is not really going to do us a whole lot of good if we want to have significant functional muscle activity. If you want to lift weights or lift a box or move furniture or things like that, you are going to need something much more sustainable and much stronger than just a single muscle twitch. We want the muscle to stay contracted for a good long while. So rather than just having one action potential fired through a somatic motor neuron, we want a steady frequency of action potentials fired along the axon of that somatic motor neuron. Remember, we talked about how the strength of a stimulus has to do with how frequently those action potentials are coming. The more frequently those action potentials are coming through the somatic motor neuron, the more and more time the muscle fibers will stay contracted. In fact, the higher the frequency of the action potentials, the more those twitches start to sum together, and we get more and more strength and more tension in the muscle fiber. The maximum we can get is called tetanus. Basically, we reach the maximum in terms of the frequency of action potentials conducted through the axon of the somatic motor neuron, and the muscle fiber essentially spends all of its time contracted and none of its time relaxing. So we call that state of maximal tension tetanus. Okay, so now we're going to have our big discussion on muscle fatigue, which is going to help with your lab that you're doing this week. So what you're looking at here is kind of just what we discussed. You use all of your central nervous system, you get as many motor units firing as possible, and you get as many action potentials conducted through those axons as you possibly can until you reach complete tetanus. So imagine that you're getting a 50-pound dumbbell and you're holding it kind of like this. You're just holding it like that. You're using all of your muscle and all of your strength to hold it like that. And you're going to hold it there for as long as you possibly can. Well, from your central nervous system's perspective, you are focused on keeping it there, right? So the frequency of action potentials is not really a problem. You can hold that for a little while. But if you look here, after a while, the tension in the muscle starts to wane quite a lot, even if you are still trying to keep those muscles contracted. So what you're experiencing there is something that we're all familiar with, right? So muscle fatigue, the idea that over the course of time, the muscle becomes less and less able to generate the force that it originally had at tetanus. And we understand this is reversible, right? So this is something that you can fix with rest after you have strained your muscle. So what we want to do here, we're all kind of familiar with what it feels like and how it occurs, the sorts of things that you have to do in order to fatigue your muscles. But based on all the discussions that we had, especially those pertaining to excitation-contraction coupling, we want to understand exactly why a muscle fiber would become fatigued and why, another discussion that we'll be having here in a little bit, why certain muscle fibers fatigue faster or slower than others. So the first thing that we want to discuss is the muscle fiber action potential. So think about how a neuron undergoes an action potential, right? So you reach a threshold, you open sodium channels, you open potassium channels. Those potassium channels are slow to close, meaning eventually that neuron is going to get a little bit hyperpolarized, right? Remember that? So muscle fibers are very much the same way. If you keep a muscle fiber excited for too long, there's going to be an accumulation of potassium in the extracellular fluid. So this is, you can either call it impairment of the muscle fiber's excitability, or you could just say the muscle fiber is hyperpolarized. And just as the case was with a neuron, it's going to take an unusually strong EPSP, an unusually strong depolarizing stimulus, in order to get you back up to threshold. So if you hyperpolarize your muscle fibers enough, you're just not going to be able to get them to contract again until they've had a chance to rest. So that is one possible explanation for why a muscle fiber would fatigue. But as it turns out, it is not the major and most likely explanation for why muscles would get tired. So really all of the possible explanations, and keep in mind, it sounds weird to say, muscle fatigue is not something that has really been scientifically settled. There's actually quite a lot of debate on this. But from my perspective, and I happen to agree with kind of what your book has to say about it, most cases of muscle fatigue can kind of be explained by what we have here in this chart right here. So what I just got done telling you happens at the level of excitation-contraction coupling. Basically, we hyperpolarize the muscle fiber and it becomes less able to be stimulated. Another possible explanation is that the longer we keep the muscle fiber contracted, we start to release, we start to have a deficit in calcium storage, meaning that we're not doing a good job of getting calcium to this troponin. That could be another explanation. You could be dealing with a deficit in neurotransmitter release from the somatic motor neuron, keeping in mind it doesn't have an infinite amount of acetylcholine. So if your somatic motor neuron is on for a long period of time, it might run out of acetylcholine. So that's something to keep in mind. But the major explanation, and you probably saw this coming a mile away, the muscle fiber runs out of energy, right? We even talked about that just previously. You need ATP in order to get a muscle contracted and get it back into a relaxed state. So if you're using your muscles and your demand for energy outstrips your ability to make energy, you're not going to get muscle contraction, and that would produce a very serious case of muscle fatigue. So we call these depletion theories. Whether you run out of phosphocreatine, you run low on ATP stores, you run out of glycogen, those are very likely explanations for why most cases of muscle fatigue would occur. So as we kind of finish up here on skeletal muscle, we need to be aware that there are actually several different types of skeletal muscle. So these are probably things that you've heard of before. So you've probably heard of slow twitch muscle fibers, and fast twitch muscle fibers. So, skeletal muscle fibers, as you can kind of tell from the names, they are distinguished based on how quickly they reach maximal tension. I bet you can tell which is which, right? So this one right here, A, is a fast twitch fiber. Basically, from the time it is stimulated with a somatic motor neuron, it reaches tetanus very, very quickly. The slow twitch fiber takes a little while. There's a little bit of a wind up before it really gets to maximal tension there. So just hearing that part of it, you're probably thinking, okay, well, fast twitch fibers are obviously superior, right? Well, not the case. So as is with pretty much everything, it's probably better than it sounds, right? So there's a little bit of a trade-off here. It's true that fast twitch fibers reach tetanus very quickly, but they also poop themselves out very quickly. They're highly, highly susceptible to fatigue. So that's why you see us reaching tetanus, and then you go back to the way things were pretty quickly. You just run out of energy after too long. Slow twitch fibers, it is kind of a bummer that they take a while to get going, but they are very, very resistant to fatigue. They can go for long periods of time without having to rest. So slow twitch fibers, like those that you find in your leg muscles, are able to sustain contraction for long periods of time without fatigue. But why is that? Why are slow twitch fibers less inclined to become fatigued more so than fast twitch fibers? What is the difference between the two? So keep in mind, we just got done discussing muscle fatigue, and we said the major thing that causes muscles to fatigue is that they run out of energy. So if slow twitch fibers are less susceptible to fatigue than fast twitch fibers, it has to have something to do with energy, right? So there are actually a number of factors that slow twitch fibers have in their favor that fast twitch fibers do not. Slow twitch fibers have more mitochondria, that means more ATP production. They get a more rich arterial blood supply, that's more oxygen coming in. And then they also have a very high concentration of a special molecule called myoglobin. So myoglobin, you're probably thinking, sounds an awful lot like hemoglobin. You've heard of hemoglobin before, I am sure. You understand that hemoglobin is the oxygen-carrying protein complex that is found within red blood cells, and it is responsible for delivering oxygen all over the body, right? Well, it turns out myoglobin does something very, very similar. It also binds to and carries oxygen, but it does so pretty much exclusively within slow twitch muscle fibers. In fact, this is actually the reason that slow twitch muscle appears red in color. It's because of all that myoglobin. Blood is red, slow twitch muscle fibers are red, so there you go. So because slow twitch muscle fibers have such an exorbitant ability to make ATP through aerobic respiration, through oxidative phosphorylation, we call these fibers slow oxidative fibers. So if we look at this picture right here, we can actually see the different types of muscle fibers all in one picture. So your slow twitch fibers, the slow oxidative fibers, are these dark red ones. So as we said, that dark red color is due to the high concentration of myoglobin there. So those fast twitch fibers that we looked at before, you will notice here, do not have any of that color. They pretty much appear white. So the fast glycolytic fibers, as we will call them now, because they do not have many mitochondria, they do not get a very rich blood supply, and they do not have any myoglobin, we call them fast glycolytic fibers because they are much more heavily reliant on glycolysis to make energy for themselves. And as we discussed, glycolysis does not make very much ATP, so you can definitely imagine here why it is that these fibers run out of ATP very quickly and why they fatigue so easily. So if we go backwards here, you're probably thinking, okay, we went over this tension graph here, but Dr. O really only talked about A and C. So the fast glycolytic fiber here with A and the slow oxidative fiber there with C. What's the deal with that other one? What's the deal with the one in the middle? So that is what is called a fast oxidative fiber. It is an intermediate type of fiber that basically is the best of both worlds. Essentially, it is a hybrid of both fast glycolytic and slow oxidative fibers. Hybrid in the way that it has some of the benefits of one without some of the drawbacks of the other. So essentially, a fast oxidative fiber does not have as much myoglobin as a slow oxidative fiber, but it's definitely got a heck of a lot more than the FG fiber. So what that means is that it is going to appear pink in color. So that's why you can see some of those pink FO fibers right there. Because they are intermediate, like I said, you basically get the best of both worlds. So a fast oxidative fiber will not reach maximal tension as quickly as an FG fiber, but it will not fatigue quite as easily either. It will reach maximal tension quicker than an SO fiber, but it is not quite as resistant to fatigue either. So it's just kind of a nice mix of the best of both worlds. So this table right here might be useful for some of your studying purposes, just in terms of comparing and contrasting the three types of muscle fibers. You don't have to pay attention to every single little thing here. For example, you can skip over myosin, ATPase activity, diameter. Basically, the things I would focus on here are the things that have something to do with energy. So the color, which we related to myoglobin, the mitochondria, which we know make ATP, capillary density, which tells us how much oxygen from the blood is being delivered. So those are the things that I would definitely pay attention to there. So this is an interesting looking graph, and we are just going to take this as a chance to do a little real world introspection on different types of people and who is good at doing different types of athletic activities and how that relates to the types of muscle fibers they are going to rely on. So there's a lot of information in this graph. We are going to focus on two particular types of individuals here: the world-class sprinter and the world-class marathon runner. So in order to really appreciate the differences here, we need to know exactly what types of activities these people are doing. So let's talk about a sprint first of all. A sprint is a very short distance race. So a sprint can be anything from 50 meters, which is a race that, if you're a world-class athlete, should be over in about 5 seconds, to 100 meters, that's about 10 seconds, to 200 meters, that's about 19 to 20 seconds. So these are races that should not last very long. So to begin with, if you're running any kind of race, your natural inclination is to think, okay, I want the fast twitch muscle fibers. I want those muscle fibers to get up to maximal tension as soon as they possibly can. But then you remember the drawback, right? You remember that, okay, but these muscle fibers are going to get fatigued very quickly. Well, that should not matter too much if we're running a very short race. We get those muscle fibers up to maximal tension right away. We run as fast as we possibly can. We get to the finish line in 10 to 20 seconds before the fatigue ever kicks in. We'll deal with the fatigue after we've already got a gold medal around our neck, right? So people who are world-class sprinters, as you can see from this graph, tend to rely much more heavily on fast twitch fibers. Slow twitch fibers would not do them very good because, as we said, it takes them a long time to get up to maximal tension, and it's such a short race that the resistance to fatigue doesn't really do you any good in the long run anyway. Okay, but what about a marathon? A marathon is definitely not a short distance race. We're talking 26.2 miles. For a world-class marathon runner, you're really talking about finishing that in anywhere between 2 and 3 hours. So yeah, definitely not a short race. So, as you can imagine, these people are going to much more heavily rely on slow twitch fibers. If you try to run a marathon with fast twitch fibers, you might get off to a fantastic start. You might sprint ahead to the head of the pack, but you're going to absolutely wear yourself out in a matter of minutes with hours left to go in the race. And other people are going to run past you, and it's all going to be over for you at that point. So for something like a marathon, you really want to rely on your slow twitch fibers. Basically, you're kind of taking it easy. You're counting on the fact that they are resistant to fatigue, and you want that to carry you through the race. So hopefully you found that interesting, and something else you will hopefully find interesting if you kind of look through this on your own. This table lists some of the benefits that you get from endurance training, basically all the stuff that you do to try to get yourself into shape, like going to the gym, running the treadmill, and things like that. But what this is kind of letting you know is how some of those benefits actually cause your muscle fibers to become more resistant to fatigue. So take a look at this on your own, but definitely keep in mind that all of these in some way are going to have something to do with the muscle fatigue that we talked about, particularly making more and more energy for yourself if you're the muscle fiber. Okay, so we are almost done with chapter 10 at this point. Recall, we're going to spend most of our time here talking about skeletal muscle with the idea that cardiac and smooth muscle, cardiac more so than smooth muscle, are really not that different. So a lot of the excitation-contraction coupling stuff, the action potential stuff that we talked about, it's not really going to be that different here, at least not different enough to warrant us really spending a whole lot of time talking about it. So all of the stuff that we talked about skeletal muscle, unless I say differently, that's going to be all you have to know. You can just kind of assume that at least something similar occurs with cardiac and smooth muscle. Again, unless I say differently. And we will say differently here in just a second here for a few things. So let's just say a couple of words about cardiac muscle and smooth muscle. We'll start with cardiac muscle here, and keep in mind, I'm not shortchanging you with cardiac muscle. We're about to start 3 straight chapters on cardiovascular and circulatory physiology, so you're definitely going to get plenty of this. But let's just have a quick little overview, a quick little preamble talking about cardiac muscle. So as you recall, an individual cardiac muscle fiber is called a cardiomyocyte, and just as the case was with skeletal muscle, these guys are striated too. That's actually great news for us because previously in part 1 of this lecture, we learned what makes a skeletal muscle striated. It's actin and myosin organized into sarcomeres, right? So guess what we can say about cardiac muscle here? If it is striated, it must have actin and myosin organized into sarcomeres. So the good news here is that the sliding filament contraction mechanism works almost exactly the same in cardiac muscle as it does in skeletal muscle. So we really don't need to learn a whole new mechanism just because we've moved to a different cell type. So that's great news for us. But something that is very different here is the presence of those intercalated discs that we've talked about. So the intercalated discs mainly contain a type of cell-to-cell junction called a gap junction. Think of a gap junction kind of like a hollow tunnel that joins the sarcoplasm of one cell to the sarcoplasm of another. So what that means is that any materials that can fit into that tunnel can pass from the sarcoplasm of one cell into the next. Mainly what we're talking about here are ions, sodium, potassium, calcium. Ions can move from cell to cell through these gap junctions, and because we have already over time come to appreciate the role that ions play in electrical stimulation of excitable cells, this tells us that these cells are what we're going to call electrically synapsed. So we'll understand what the implication of that is more fully in chapter 18, but for now, imagine that this cell here, this cardiomyocyte in the top left, say it becomes very heavily depolarized, so it has a whole bunch of sodium inside the sarcoplasm. Well, guess what? That sodium can actually move freely through those gap junctions and then depolarize the cell right next door. So ions and depolarization or hyperpolarization can actually spread from cell to cell to cell, provided that those cells are joined by gap junctions. And then these intercalated discs also contain another type of junction called a desmosome, but those are not really important to electrical synapsing, so don't worry about those too much. So just to kind of reiterate what I said on the previous slide, because those ions can move freely from cell to cell through gap junctions, any depolarization or action potential or whatever else that is produced in one cardiomyocyte can actually spread throughout the cardiac tissue, provided that all of those cells are electrically synapsed with each other through gap junctions. So again, just imagine that you have tunnels that link cell to cell to cell, tunnels that go through both sets of plasma membranes. So this is going to become important for us when we really start talking about the contraction of the heart. So it turns out that all the cardiomyocytes in the heart are not actually connected to each other. So cardiomyocytes are actually connected to each other in units. Kind of think of them as islands. You have one island that is separated from other islands. All the cardiomyocytes on one island are connected to each other through gap junctions, but they are not connected to the cardiomyocytes of another island. These islands that I'm talking about are called myocardia, One myocardium consists of a big group of cardiomyocytes that are close by and all connected through gap junctions. So just because you get depolarization, an action potential, and contraction of one myocardium does not necessarily mean a different and separate myocardium is going to contract as well. So we will have a more complete discussion on that in chapter 18, but that's something to think about for now. Another big difference between skeletal and cardiac muscle is where do we get our stimulation? Where do we get our action potentials? Skeletal muscle, being voluntarily contracted tissue, gets its stimulation from somatic motor neurons. We've actually already had a discussion about how skeletal muscle will not contract unless a somatic motor neuron tells it so, right? So cardiac muscle, as we've already said, is innervated by sympathetic and parasympathetic postganglionic neurons, right? But here's the deal. The heart will continue to contract regardless of whether those neurons are there or not. So the heart, cardiac muscle, is not contracting because the nervous system is telling it to. We've already said all the autonomic nervous system can do is either speed the heart up or slow it down, make it work harder or make it work less hard. So what actually causes the heart to contract is a section of tissue within the heart, different sections of tissue which are electrically active, called pacemaker regions. So the pacemaker region that we're looking at here, located just above the right atrium, which is called the sinoatrial node, will produce waves of depolarization, one right after another, in rhythmic fashion. And it will do this no matter what, unless there's some kind of disease-ridden defect with it. So these potentials are called pacemaker potentials. And as you can see here, the depolarization we get from these pacemaker potentials can spread throughout the myocardia. So because of the presence of those gap junctions, we can excite the cardiomyocytes within different myocardia over and over and over again. And as we are going to see in chapter 18, this is what's going to be responsible for the fact that our heart beats at a certain rate or rhythm. Normally, heart rate is supposed to be about 60 beats per minute. So if we get that depolarization happening at about that rate, that'll be about how many times our heart beats a minute. So let's finish things off by just having a very, very brief discussion on smooth muscle. So smooth muscle, I've said it before, it's about as different as compared to the other two types of muscle as humanly possible. It's similar to cardiac muscle in that it is also a target of the autonomic nervous system. It's also completely involuntary, so it's autonomically controlled, but it's not striated. So what that tells us is that we don't have actin and myosin organized in a sarcomere setting. There is actin and myosin, so we don't need to worry about that. There is actin and myosin, but the organization is totally different. So we don't have those alternating light and dark bands, which is why we call it smooth muscle. It's not striated. So smooth muscle can be arranged in one of two different ways. Here, in this picture, you are looking at the smooth muscle that is found in the middlemost layer of most arteries, veins, and arterioles, and we'll learn more about that in chapter 19. But here you can see smooth muscle arranged in a circular fashion, and we've already talked about how the sympathetic nervous system can cause the contraction and dilation of these muscles in order to promote or restrict blood flow to other places. So don't forget, this is only the sympathetic nervous system that can do this, not the parasympathetic. So most of this is coming from either norepinephrine from sympathetic postganglionic neurons or epinephrine from the adrenal medulla. Another place that you can see circular smooth muscle is in the iris of your eye. We talked about how that acts kind of like a diaphragm to regulate how much light can enter into the retina. Alternatively, smooth muscle can be arranged kind of as flat sheets in what we call longitudinal fashion. So you typically see that in food pathways like the esophagus and then different places in the digestive tract. Okay, so smooth muscle is weird, right? Smooth muscle is weird. So what we're looking at here is we're looking at the organization of actin and myosin thick and thin filaments here in smooth muscle. And it's weird. I'm just never going to get over how weird it is. So we already said that the organization is not in sarcomeres. So what that means is we don't have any Z discs. So no Z discs anchoring down the thin filaments. So what we see here is that we have, instead of Z discs, we have these dense bodies. So these dense bodies that are either located in the cytoplasm or are attached to the innermost layer of the sarcolemma. So these dense bodies are going to anchor down the thin filaments, and then you'll still have some thick filaments kind of wedged in between. So you'll get an appreciation for what this contraction looks like here in just a minute, but it's going to be a little bit funky looking, just to let you know. So one question you might be having is, why organize the thick and thin filaments like this? And the reason for it is because smooth muscle has to be able to undergo stretching forces much more so than cardiac and skeletal muscle. So it gets back to that elasticity and extensibility issue that we talked about at the beginning of the chapter. So basically, if you have striated muscle, in which case you've got your thick and your thin filaments organized into sarcomeres, if these are my Z disc, if we stretch that muscle out and we lengthen the sarcomere, if we stretch that sarcomere out too much, we risk getting to the point where there is no longer any overlap between the thick and the thin filaments. And then at that point, the muscle's pretty much torn at that point, and we're going to have to repair it before it can go back to being normal. Smooth muscle is able to overcome that much more so because of the unique way the thick and the thin filaments are organized here. So here, just kind of a reminder, go back to chapter 15 if you need to for a review on autonomic postganglionic neurons and the role of varicosities in ensuring even release of neurotransmitter among the tissues. So here, our target isn't just one individual muscle fiber, it's actually many, many of them. Okay, so here you can actually see a little bit of a demonstration of what a smooth muscle fiber looks like when it contracts. So based on kind of what we know about the distribution of those dense bodies, you can kind of see here what happens to the smooth muscle fiber when it contracts. And I don't know, maybe it's just me, but it's just a little bit unsettling looking. It almost looks like something out of a horror movie, but best not to think about it. I don't want to put any bad ideas in your all's head, but it's weird. So if I ask a question on the exam, what are smooth muscle fibers, be looking out for the word weird, right? No, I'm just kidding. Okay, and then this is the last slide right here. Basically just another study aid for you, just kind of an exhaustive comparison and contrasting of the three different types of muscle fibers that we've talked about. Keep in mind, as it pertains to excitation-contraction coupling, there are some differences that we didn't really talk about. For example, smooth muscle does not have a troponin, and it instead has a different calcium-binding protein called calmodulin. You don't need to worry about that. So there are a couple differences here and there, so try not to get too hung up on that. Just kind of make sure that you focus on some of the things that we had talked about before. But it definitely, I think, will be worth your time to kind of bookmark this table here and use it for study purposes to make sure that you have this all down pretty solidly. Okay, so that does it for Chapter 10. So appreciated your all's attention. So especially the thing to pay attention to moving forward as we move into cardiovascular, make sure you pay attention to that last bit about cardiac muscle, because we really are going to get into a little bit more detail there. All right, so that does it for Dr. O. So I am going to sign off now. Thanks again, and I will see you next time.